everyone, so today I'm going to review protein synthesis with you. This is the process of transcription and translation. You've already seen all of this, but I just wanted to take a moment to go over everything and make sure I answer any of your questions and hopefully help you to figure this out. It can be a pretty complex process, but I think once you get the hang of it, it becomes pretty easy to wrap your head around it. So remember that this whole process, both transcription and translation occur in order to make a protein. Back when we learned about the biomolecules, we learned that proteins carry out a whole range of functions in the body. It was almost a joke. What was the function of a protein? You could say everything, right? Enzymes are proteins. Antibodies are proteins, which you might be hearing about in the news that we're doing antibody testing for the coronavirus. That's a protein. Collagen is a protein. Insulin is a protein. Keratin is a protein. Every function that has to be carried out in the body is done by a protein. We've already actually learned about a ton of proteins. We learned about ATP synthase, which makes ATP during cellular respiration. We learned about helicase, which is an enzyme that unzips DNA during DNA replication. We learned about all sorts of things, but those are just a few off the top of my head. But we also learned that the instructions for building proteins come from nucleic acids. And specifically, the instructions are stored in your DNA. So the reason you look and act and behave the way you do is because you have a certain set of genes that gives a certain set of instructions for building your proteins. And then those proteins carry out the functions of your body. So it is because of your DNA making those proteins, and then the proteins do everything in your body, that we say, your genes create you. And so we're going to talk about these two processes, transcription and translation, to see how you go from DNA to protein. All right? So I'm just going to start with a DNA molecule. Okay? Just a random string of DNA. Remember that DNA is double-stranded. So I'm just going to let me just make sure I set myself up right here for later on. Okay, so that's one side. And then our other side would be complementary, following the base pairing rules. And this is DNA. So I'm still adding my thymine. Okay, so that's going to be our strand of DNA, all right? And so this is not DNA replication. DNA replication we learned many weeks ago now, at the beginning of remote learning. That's when helicase comes in, DNA polymerase, and semi-conservative replication, right? DNA replication occurs during S phase of interphase to make copies of chromosomes. That's a completely different process than what we're doing here. That was to prepare for cell division. This is to make proteins. Two entirely different processes. Similar in ways, but different purposes, okay? So this is our DNA molecule. So I'm first going to show you how you can transcribe and translate this gene, and then I'll get a little bit more into the particulars of the process. So when we're talking about transcription, we give the DNA molecule, the two strands, different names. I'm going to label this as the template strand and this as the coding strand, okay? The first thing that RNA polymerase looks for is the promoter, okay? RNA polymerase is the enzyme that carries out this whole process, and the first thing it looks for is a promoter. That tells RNA polymerase where to start reading the DNA. It essentially marks the beginning of a gene. One gene matches one protein. So this sequence of DNA right here would contain a gene, and that gene gives the instructions for building a protein. One gene matches one protein. So let's say you have 200 different proteins in your body. You would have 2 different genes in your genome, your entire DNA profile, okay? You actually have way more than that, but that's just an example. So this sequence, let's say, gives the instructions for building insulin, which is a protein involved in sugar metabolism. A different gene would give instructions for building keratin, which is the protein that makes up your hair and nails, okay? So what the promoter does is it basically signals the beginning of a gene, okay? Not all of your DNA codes for proteins. So there are letters in your DNA that are not part of a gene. The promoter is one of them. The promoter isn't actually part of the gene. It is a little bit before the gene, and it tells RNA polymerase where to start reading. There's all sorts of what we call junk DNA. That's actually what the scientific term is. It's called junk DNA. That doesn't code for proteins, but it has some kind of purpose. It might be leftover DNA from your ancestors, right? Once upon a time, cavemen or even earlier ancestors needed genes for tails, needed genes for a hairier body, needed genes for, why can't I think of anything else? Needed genes for longer canine teeth, right? That DNA might still be in your genome, it's just not used anymore. So there might be something like that. There's also regulatory sequences. There's all sorts of junk DNA. But what we're concerned about in transcription is the gene, okay? So I'm going to say that these 5 letters right here are the promoter, okay? So what RNA polymerase does is it scans the DNA for that promoter and attaches to it, okay? That tells RNA polymerase where to start reading, and it marks this as the beginning of the gene, okay? The way you know that, so let's say I didn't label those 2 strands in the beginning. The way you know which is the template strand and which is the coding strand is that the template strand has the promoter. The template strand is the strand that RNA polymerase reads. Okay, RNA polymerase does not read both strands. It's only going to read one strand of DNA, and it reads the template strand, which contains the promoter. Okay, so RNA polymerase attaches here and then starts reading. So I'm going to start transcribing from here, and I'm going to end here. Okay, so actually, you know what? I'm going to add a couple letters down here. I wasn't thinking ahead when I wrote these letters. Whenever you make up a DNA sequence, you have to think ahead to translation to see what it's going to look like. So I want to make sure, yeah, see, I would have messed up. So let me just make sure I set myself up here for success later on. Okay, and we'll be good. Sorry about that. All right, again, this was the template strand. This is the coding strand. We can completely ignore the coding strand. That is not used in transcription. So we only care about transcribing the template strand. So I'm going to start with a T. When I see a T, it's an A. When I see an A, it's a U. Because now this is mRNA. And RNA, all types of RNA, do not include thymine. They substitute uracil. Okay? Again, I've realized that I wait. Oh no, we'll be okay. Sorry. Always got to think ahead with these. Okay. G, C, C. Of course, you won't have to think ahead because the questions will already be made for you. But if you're making these up on the fly, you have to be very careful about what letters you choose. Okay. So now this is our gene. Okay. Let's say this isn't even, let's say this is transcription in a flower. Plants do this process as well. Let's say that this is the gene for flower color. Okay. And we don't know what the protein is going to look like yet. Based on just this strand of DNA, we need to see what the amino acids are going to be to figure out what color the flower is going to be. But regardless, this is the gene right here that codes for flower color. In reality, genes are much larger. They're thousands of base pairs long, but for simplicity, we've shortened it. But you get the idea. So there's something important that happens after transcription but before translation, and it's called post-transcriptional processing. You read about it a little bit in the transcription slideshow. There's a couple things that happen. So the first thing that happens is that introns get cut out. Introns are sections of that junk DNA that are contained in the gene. But if they were left in there and were involved in translation, the resulting protein would be incorrect. So sometimes there might be leftover sequences or leftover letters from your ancestors. I can't think of any examples off the top of my head, but introns are little tiny chunks of that junk DNA that isn't needed anymore. And it needs to be cut out of the gene, because if those letters were left in the mRNA, they would get translated and would become part of the protein. But they're not supposed to be there anymore. They're still there. They're just kind of relics from your ancestors. You've evolved since that those letters were needed, and they have to be cut out. So those are called introns. Introns need to be cut out. I know it doesn't sound right? It makes more sense that exons would be cut out, but it's the other way around. So I'm just going to pick, again, let me make sure. Okay, I'm going to cut out this. I'm going to say that right here, this is the intron. So this is really called pre-mRNA. And then your final mRNA just literally has that cut out. So it would be A, U, G, C, G, U, 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 U. Skip that. And then we just go to C, C, G, U, A, G, C, C, U. Okay? So this would be then called an exon. And this would be called an exon. And those get spliced together. So the intron gets cut out and the exons get spliced together. Now, I don't know if you read about this in the presentation. I don't think it was in there. But something else also happens to the mRNA. It gets two things added onto each end. It gets what's called a 5' cap and a poly A tail, literally just a tail of adenines. Okay, so that's added to the pre-mRNA and it stays in the final mRNA. 5' cap, 5' just relates to the labeling scheme given to DNA. DNA is given a 5' end and a 3' end. On the 5' end, methyl G cap is added. You don't need to know that name. It's a methylated guanine. It's just a chemical group that gets added onto this end. And then to this end, a bunch of adenines get added. And this is basically like armor. This mRNA is about to leave the nucleus and go out into the cytoplasm. And that cytoplasm is an unsafe place for nucleic acids, right? Nucleic acids usually are contained in the nucleus. They're safe there. That's where the DNA is. DNA never gets degraded. It's always in the nucleus. It's always safe. This RNA is about to leave the nucleus where it's safe and go out into the cytoplasm where it doesn't normally go, right? So it adds this 5' cap and this poly A tail. It's like armor. It's to help protect the important part. We need this part, right? This is going to be the stuff that gets degraded first. And so this gets added on as kind of a, the point of first attack. This is what gets attacked first so that this is maintained for as long as possible, okay? So this is what leaves the nucleus. Messenger RNA, mRNA. This RNA is carrying the message from the DNA to the ribosomes in the cytoplasm because the DNA can't leave the nucleus. Okay, if it left the nucleus, it would get degraded and your cells would have no instructions anymore. It's like trying to build a house without a blueprint. Okay, so now this whole thing leaves the nucleus through a nuclear pore and goes and attaches to a ribosome. So now we're going to do translation. That was transcription right here. Okay. This is post-transcriptional processing. And now we're going to go into translation. So translation is the final step. This is carried out by a ribosome. If you remember when we learned about all the organelles, we learned that ribosomes make proteins. They build proteins. Now we're seeing what they actually do to do that. Okay? So the ribosome starts at what we call the start codon. The start codon is AUG. Okay? Even if there were other letters here, let's say, pretend that there were other letters here. Okay? The ribosome completely ignores those letters and will always start at AUG. Okay? It's kind of like reading a book, but you have some, quirk that you only start reading that book at the first, let's say you don't start reading until you find the word yellow. That might be on page 600, but that is where you start reading. That's kind of what the ribosome does. It will ignore every letter above this start codon here, it starts reading at AUG, and that is always the case. Okay, ribosomes always start at AUG. Sometimes if you're doing practice problems like this, the question will try to trick you and will put letters before this, and a lot of students fall into the trap of doing codons that come before AUG, but you ignore everything before the AUG and start reading here. Okay, so your first codon is AUG, your second codon is CGU, third is UUU. Remember, codons are every 3 letters, so I'm just going along here, and those are our codons. Okay, and this is the start codon. So ribosomes read the mRNA 3 letters at a time. It's kind of like a sentence. This is the whole sentence, and these are words, right? We don't read sentences letter by letter. We also don't read sentences, 3 words at a time. We read 1 word at a time. So this is a word, this is a word. But for ribosomes, every word just happens to be 3 letters long. So if this was a sentence, this sentence would have 1, 2, 3, 4, 5, 6 words and a total of 18 letters. Okay, these aren't codons. This was the poly A tail that got added for protection. The ribosome doesn't read that. Okay, so it starts with AUG, and this is when you pull out your handy dandy codon chart. Okay, the cell doesn't need this. The cell knows how to convert from RNA to protein. Remember, these are completely different molecules. We learned about 4 macromolecules when we learned about, in our biochemistry unit, we learned about proteins, nucleic acids, carbohydrates, and lipids. mRNA is a nucleic acid, while proteins are proteins. They're completely different languages, right? This is made up of nucleotides, adenine, uracil, guanine, right? These are nucleotides. What are proteins made up of? Amino acids, an entirely different language. Those are different words, different letters, okay? The cell knows how to translate between those two. That's why this process is called translation. You're going from one language to another. We don't. We need this. This is like our 2-language dictionary. Imagine you're traveling to a different country. Have this with you at all times. If you want to print one out from online, and just keep that with you throughout this unit, it'll probably be a good idea because you depend on this so much. There's no way you can memorize this, okay? So this is your tool for translation. So now this is going to be what we call a polypeptide, another name for a protein, okay? So AUG, you start at the middle, AUG. Start with the biggest, go to the next row, go to the last row, gives you methionine. And you can do abbreviations. The abbreviation is written right here. But be careful, it's not always the first 3 letters. For example, glutamine, the abbreviation is GLN, not the first 3 letters. So just be careful with that. And because this is always the codon you start with, methionine is always the first amino acid in a polypeptide, okay? CGU. CGU gives you arginine, ARG. UUU. UUU gives you phenylalanine, PHE. CCG. CCG gives you proline, PRO. UAG. UAG is a stop codon. Now, some teachers don't like you to write this because this is misleading. If this was a string of amino acids, this kind of makes it look like there's another amino acid here. But there's no such thing as stapamine, right? This is not an amino acid. This is the polypeptide. It stopped here. There's no more amino acids after that. But if you just for yourself want to write stop, that's okay, okay? Just know that if you go on to other science classes, this might be considered incorrect because it is misleading. It makes it look like there are 1, 2, 3, 4, 5 amino acids in this chain when really there's nothing here, okay? And what does stop mean? It means stop. One of the most common mistakes is that students will then go on to do the next codon. CCU is proline again. Stop means stop. The ribosome stops reading and goes to find a new mRNA, okay? So once the ribosome that is reading these codons reaches a stop codon, it stops. Translation stops, and this is your final polypeptide. This is your protein. This is what you intended to make. This is protein synthesis. You went from DNA to a protein, all right? Now remember, I said we'll pretend that this is a flower gene. And we were going to say it was the gene for flower color. Now let's say that there are 3 alleles for flower color. The flower color could be pink, blue, or purple, okay? Let's say then that this particular sequence of amino acids makes the flower purple. If, let's say, you had gotten methionine, arginine, phenylalanine, cysteine. Sometimes I feel like I make these up. Nope, cysteine is one of them. And then stop. Maybe this gives you pink flowers. So it's a minor difference, but it's enough that it's going to change the shape of the protein. And that shape of the protein is going to determine the function. It's going to create a different pigment. And the flower will be a different color. And to get cysteine, this codon would have had to be different. To get cysteine, this codon would have had to be UGC or UGU. You have 2 options, okay? So that's why the sequence of the DNA ultimately determines the sequence of the amino acids. It's my cat. He's coming to say hello. Ultimately determines, he's going to jump up here. Ultimately determines the shape of the protein and its overall function, okay? So once the cell gets to this step, once it gets a polypeptide, that polypeptide folds into a 3D structure and becomes the proteins that we have come to learn about, those enzymes, those antibodies, all of those things, okay? Now I just want to take a moment and backtrack. Let me erase this real quick. So there's a lot that goes on during translation, and I just want to take a moment to talk about that. So let's say that we have our, I'm going to ignore all this and start from a different mRNA sequence. So this is going to be my mRNA. Remember, it's going to have that 5' cap. And then I'm just going to do, again, random string of letters. Okay, and then we have our poly A tail. So I'm going to go into a little bit about the actual process of translation. How does the cell go from one language to another? So again, a ribosome attaches. Where does it attach? It attaches to the start codon. So I would not start transcribing at this codon. This is not the first codon. This is the first codon. And a good trick is to always separate your codons, because, you know, your eyes will play tricks on you, and you could possibly skip a letter or whatever. Some people like to put lines in between, whatever you feel comfortable doing, just so that you, get comfortable doing this. So the ribosome is going to attach here. The ribosome is a 2-piece structure. It's got 2 subunits, and it starts reading. And what it actually does is it brings in something called tRNA. So this is mRNA. Ribosomes are actually made up of something called rRNA. And the rRNA is reading the codons and is bringing in what we call tRNA. tRNA stands for transfer RNA. That tRNA, I'm going to actually make it a T shape, carries with it an amino acid and what we call an anticodon. The anticodon is complementary to the codon. So what would the anticodon be of this tRNA? If this is AUG, and I said that the anticodon is complementary, what would the anticodon be? It would be UAC. Remember, this is tRNA. RNA always has uracil. Okay? And then it's also carrying an amino acid. We know that AUG, according to our chart, is going to be carrying methionine. Okay? So the ribosome brings in this tRNA and actually has it match up with the codon. So there's complementary matching between this codon and this anticodon. That is how this tRNA knows to come here, right? It does not, it's not like a ribosome has a brain or even has, a complex computer to know to go find this tRNA. The reason this tRNA comes here is because it has this anticodon, and this is just chemistry, right? These letters form bonds. And whenever this tRNA has this anticodon, it will always be attached to methionine. There are enzymes in the cell that bring the correct amino acid to the right tRNA, so that this tRNA with this anticodon will always have this amino acid. Same thing, so now the ribosome is going to work its way down. Okay. And it's going to go to the next codon. So now the ribosome is here. It reads GCA. It's going to bring in a tRNA with the anticodon CGU. Now, this tRNA will always be attached to the matching amino acid. The cell, like I said, has enzymes that attach the right amino acid to any tRNA that has this anticodon. But we need this. Now make sure that when you're reading this, this is a codon chart. Make sure that when you're using this, you're reading the codon and not the anticodon. Let me show you why. So we're reading the codon. We want GCA, right? This is our mRNA that we're reading. We want GCA. GCA gives you alanine. If we had used the anticodon, CGU, gives you arginine, a completely different amino acid that will completely change the shape of the amino acid, that'll completely change the shape of the protein. Remember that structure determines function. If that protein has an incorrect amino acid, it won't have the right shape and it won't work correctly. Just think about enzymes. If you remember when we learned about enzymes, we learned that they have that like Pac-Man shape. This is the enzyme, which is a protein made up of amino acids, and it binds to a substrate that has to have a very specific shape. So if this enzyme has an incorrect amino acid, then it will have a different shape. And this substrate doesn't fit perfectly anymore. Okay? So just something to keep in mind. All right? So make sure you're reading the codons. and CGU. Oh, I even made the mistake. You need GCA. Make sure you're doing the right one. GCA, alanine. Now, before, when I took you through the process of transcribing and translating a gene, I ignored the tRNA, right? We have this. We can directly go from codon to amino acid. The cell cannot. The cell cannot jump from mRNA codon directly to amino acid. It needs that tRNA as the go-between. I'm not going to do all the rest, but you get the idea. This is your stop codon. I'm not going to show the ribosome moving. I'll do the rest of these. ACA gives you lysine, and that would have its own tRNA and an anticodon and all that. GGG gives you glycine. UCA gives you serine. And UAA is stop. Okay, so this is your chain of amino acids. This goes and then folds to make a 3D structure, and you get your protein. A protein has to have a very specific shape in order to do its function, and it gets its shape by having the right amino acids. It gets the right amino acid by reading the DNA. Okay, so this is the process of protein synthesis. You've seen all of this in, the practice work I've given you, the notes, the slideshows, but I just wanted to make sure that I personally went through it with you. If you have any questions, please reach out. I know that this is complex. One suggestion I have is to go watch videos. If you're stuck on this, if it doesn't really click, watch a video to see the moving parts. Right, it's hard to really visualize this on a 2D surface, but seeing it 3D is really helpful. Actually, at the end of the translation presentation that you saw last week, those set of slides where you wrote you typed the notes into the document, there was those slides that went along with it. At the very end of those slides, in the last slide, there's a link to a video. If you didn't watch that already, go watch it. And if you already did, I suggest going to watch it again because it's a fabulous video. And it really does a great job of showing you how DNA gives instructions for making proteins. All right? So hope this helped anyone who was stuck. Please reach out with any questions and enjoy the rest of your day.